Hey everybody, welcome back to Microbiology. Today we're going to go through the bacterial genetics. So the bacterial genome is a circular haploid DNA in a supercoiled form, and there are no introns and no nuclear membrane. Instead, it's kind of floating around in something called a nucleoid. The plasmid is an extra genetic material, and sometimes that can be incorporated into the main chromosome, especially when things are like being passed from one bacteria to another. And if it's able to do that, it's called something called an episome. Within the genome is the DNA gyrase, topoisomerase, and these are responsible for the unwinding of the DNA. And these are targeted by the antibacterial agent chloroquinone. This is really effective against malaria, actually. And there is something called a prophage, a bacteriophage gene. So a bacteriophage is a virus that can actually infect the bacterium. So replication is basically the same as a eukaryotic DNA in some respects. The other, the major difference is that the enzymes and the proteins are different. So those are going to be targeted by the specific antibiotics, such as fluoroquinolones, and those bind to the DNA gyrase, and those inhibit the uncoiling or supercoiling. So there's usually like a single-stranded nick. There's a displacement of the nick strand, and then it's going to be rejoined here, and then the nicked strand is going to be used as a template to make the DNA of the new daughter bacteria. So the synthesis of the second strand requires like other enzymes that aren't really like, host enzymes. That's why in replication, bacteria has to code for its own genome. So first, there's the leading strand, and then there's a nicking, and then the DNA will unwind, and then there is elongation of the new leading strand, and that will like kind of allow the displacement of this strand right here. So there's no one time of the original DNA to be like only single stranded, right? So once this finishes, it's kind of like a chain, right? This pops off, this is going to be made, so it's kind of continuous until it reaches some kind of like stop codon, and then it's terminated, and the single strand is detached, and then it uses the protein that the enzymes that the the bacteria genome coded for in order to make its new progeny. So they transfer information using a sex pili, and some bacteria, others use like operons. So these are clusters of co-regulated genes. These have a promoter, operator, and structural gene all kind of included. So these can be inducible or repressible. One of the important inducible examples are the lac operons. So these only turn on in the presence of lactose. There's something called a sigma factor, where the protein for the initiation of RNA synthesis turns on or off the gene. So this will code for whether the bacteria needs the bacterial proteins or not. The sigma factor will initiate the RNA synthesis for the bacterial products. Quorum sensing is a unique feature of the bacteria where it can generate the expression depending on the population size. So the population is too big for the host cell to be able to make enough products or proteins for the bacteria. It will kind of inhibit replication. So it depends on the amount of resources. In translation, there are unique microbial initiation, elongation, and termination factors. One of the first amino acid is going to be N-formylmethionine, or F-met, and that's going to kind of like code for the start of everything. So it codes for the soluble PAMP, which are going to be recognized by innate immunity cells as foreign material, and then more importantly, translation is going to be targeted by the antibiotics tetracycline and macrolides. So the initiation starts with the F-met when the transitional complex forms and binds. And then there's elongation, then termination, and it kind of chugs along until it hits a stop codon. Okay, so the genetic information of bacterium will change because the mutation rate is so high and expressed very quickly because of the haploid DNA. So there's no recessive or dominant modes of inheritance. Instead, there's no survival of the fittest, basically. The mutation is going to be spontaneous or induced. And the most common mutation are like the point mutations. So transition is when the one purine is going to be subbed for another purine, one pyrimidine replaced with another pyrimidine. And all of these mutations can result in either silent frameshift, multi-site mutations, helix distortion, or suppression mutation. So transformation is one of the most important mechanisms of genetic transfer in the gram-positive microorganism. The cells must have something called a competence factor, and sometimes they use the plasmids to transmit the information. So the bacteria dies, and the free DNA is going to be released, and the other related bacteria takes up the free DNA. So it's kind of like if a strep bacteria dies and releases its DNA, other strep bacteria will take it up. The uptaken DNA is going to be very sensitive to nucleases, so those can be broken up. This is when the bacteria is most susceptible to our immune system. So if taken up properly, there's crossover or homologous recombination. So here it's our DNA fragments, takes it up, and then it either gets integrated or it's degraded. For plasmid, it can just take up the plasmid. Some of them, if they're in the episome, it gets integrated. Otherwise, they just kind of keep it around. Okay, so conjugation is when the pili comes into use. It's in gram-negative. So before in the transformation, it's more for the gram-positive bacteria. So the F factor is going to be transmitted. So it's a plasmid, it's going to be transmitted. It's done using the transfer operons, and the fertility factor is going to go from one F plus plasmid to an F minus plasmid, F plus bacteria to an F minus bacteria. So the plasmid is going to be transferred over, and the F minus bacteria is going to become F plus bacteria. So they get the new plasmid, but these plasmids are not episomes, so they're not going to be integrated into the genome. So in this particular case, if this cell goes through bacterial fission, the F plasmid isn't going to be transferred. So the old donor and the new donor, if they go through binary fission, they're going to have the F plasmids, but they're going to be F minus. Oh, I'm sorry, they're not going to have the plasmids. They're just going to be F minus. The genome will be replicated, but unless the pili form and kind of like conjugates with the daughter cells, then that's how they get the F plasmid. HFR conjugation is similar, but is mediated by an HFR plasmid, which are the F plasmids that can be integrated into the host chromosomes because they contain something called an insertion sequence. So in the same way, they still need the sex pili. So first, in this particular case, the HFR cell has the integrated plasmid. So the episome is already in, then they have a connection with the sex pili, and it is going to be excised from the original and dumped into the daughter cell, or the second cell, using the sex pili. Thanks to this ORI-T enzyme, so it's going to make the nick, transfer it over. So there is no like singular plasmid, since it's already integrated into the host, the bacterial genome, right? So it's going to come over. It's a partial transfer of chromosomal DNA. The F factor, however, isn't going to be it's not going to be like transferred over, but the plasmid itself will. There's going to be receiving the double-stranded DNA, and then it's going to be integrated into the new cell. So in this case, from 3 to 6A, this only occurs if this is not like compatible with this, so it just gets degraded. In some cases, so like the F, there's Nick, this opens up, gets transferred over, and then now you have the partial chromosomal DNA. Okay, so why are the plasmids important, right? So the plasmids will carry antibiotic resistant genes. They may be successful, they may not. It's kind of like the evolutionary arms race, right? So it's just kind of useful for the bacteria to have in its back pocket. That's why it's transferred over. The virulence genes are going to be the pili genes that mediate the adherence to the epithelial cells, and it's going to help the exotoxin genes to be transferred. So why is exogenous genes important? Because the exotoxins are going to cause like the inflammation. It's going to cause the cytokine activation, stuff like that. So in transduction, there's going to be a transfer of bacterial gene information using the bacterial phage. So this needs the virus. They are the double-stranded DNA virus, and it infects the bacteria in two ways: the virulent or lytic phage, which transfers just general information. It transfers like the structural protein, the general like just virulence factor of the bacteria or virus. And in temperate or lysogenic phase, it's specialized transduction. So this is going to transduce, is going to give the new bacteria like new ways to counter antibiotic resistance. So there are the specific genes being transferred or transduced. Transposons are going to be important because they carry the insertion sequences, allowing the HFR to like transform the new cell. That's why they allow the the bacterial genome, the partial chromosomal DNA, to be transferred over using transposases, and then the resolvase is the last part of the puzzle, which inserts the partial chromosomal DNA into the bacteria's DNA. So, interestingly enough, all of these work together, so the bacteria can integrate and excise the genes and take the genes to another place while leaving itself behind. So this allows like a lot of freedom of movement for the genes. They can move to and from any DNA phage, plasmid, and bacteria genome. So that allows a huge opportunity for antimicrobial resistance like genes to be spread. So the consequences of integration: first you had to disrupt the normal gene sequence, and then you had to insert the new genes. This is problematic for the host cell. So we're not saying bacterial cell, we're saying if this bacterial cell infects the host cell. So let's say it's a cervical cell of a human. With the insertion of the new genes and stuff, it will turn off a lot of tumor repressor genes, so important genes like the P53 system, and turn on a lot of oncogenes, because these are going to be like virus factories. They're going to be trying to make more of these cells that are going to make more of the bacteria. Sorry, I keep saying virus, but I mean bacteria. So this can cause like cancer. Okay, so recombinants are going to be the integration of donor DNA into the host genome. Like I said, the homologous DNA recombination is going to occur for the exchange of DNA, and there are going to be the recombination enzymes needed. So Rec A, Rec B, etc. The site-specific recombination needs no homology, and there's just integration, there's no exchange. It requires the restriction endonucleases and the sites for the endonucleases on both DNA. So they just kind of like come in, cut things out, put things in, that's it. There's no homology needed, and it's purely integration. So the foreign DNA is going to be circular, or it can be linear, and it's going to be, the new DNA is going to be the sum of existing and the additional DNA. So let's do the, as transposonases, it's going to transport things, and then using some, we're going to use it's going to be the resolvase. So we're going to use the resolvase to insert into the genome, or the host genome, and then it's going to add in, and then you get a new DNA. Phase variation or conversion is going to be a programmed gene rearrangement. So these genes need to be on and off in order for the phenotype switching to occur. So the genes are going to be like turned on and off without regulation if phenotype switching occurs faster than the actual mutation. The transcriptional regulation of the alternative genes will occur thanks to the on-off expression, and there's no addition of genes, so this doesn't actually add to diversity. This can affect the host cell binding and the virulence, of course, and these require a promoter in order for like something to bind and be like, oh hey, this gene needs to be turned on or off. So the problem with the altered expression of the proteins, antibodies might not be effective if the target is not expressed, or it could be like less effective. That's it for now. Thank you for your attention, and good luck studying.